Welcome to Chair's Grand Rounds. I've decided to change things up a little bit today, and we're going to have two of our fundamental scientists present. I'll introduce them in turn as they speak. One of the things that people don't realize about the Department of Medicine is that as of yesterday, we had 46 PhD scientists in the department, making us one of the largest fundamental science departments in the university. And I think oftentimes we overlook some of the people who contribute to the basic science work that we do in the department. So I decided to feature two of our younger, although Dr. Rullo points out that he's not that young anymore, members of the department who are doing fundamental research. First is Dr. Josh Koenig, who we brought on faculty within the last couple of years. And some of you will have seen him featured in the media recently. Josh works in the allergy and immunology space and has done some really important work with his lab over the last couple of years and some very important publications in the recent past. And he's going to talk today about distinct phenotypes of memory B cells, hold IgE memory, and talk a bit more about the pathogenesis of allergy. He's going to talk for about 20 minutes. We'll then have a question and answer session, and then we'll move over to Dr. Rullo for his presentation about his area of research. So without further ado, Josh, why don't you fire up there?
Okay, sounds great. So these are the objectives that I put out in the mailer, and I have one conflicts report, which is in the past I received some funding from a pharmaceutical company called ALK Abelo, and a lot of the work that we did in this is in collaboration with them. So I study allergic diseases, and something I want to talk a little bit today is about how research is changing. A traditional toolkit for immunology is something that we have used to explain a lot of how the immune system works. And through this, we've understood in allergy that the pathogenesis of this disease is when people have IgE antibodies that are against a particular food. In this case, let's say peanut. When a person is exposed to peanut again, these IgE antibodies, which are loaded on the surface of these granulocytes like mast cells and basophils, they're cross-linked, so two of them come together. And when that happens, these cells will immediately release these inflammatory vasoactive mediators like histamine, platelet activating factor, leukotrienes. And this causes those immediate signs and symptoms of anaphylaxis or hives or carriaginous, these different symptoms. And so we know a lot of this because of this traditional immunology toolkit. And so what this looks like is, let's say we think IgE is at the center of this disease. So we'll go and we'll take a mouse and we'll get rid of IgE in the mouse. We'll knock it down at the genetic level. So now we have a mouse that doesn't have IgE, and we'll try and sensitize that mouse. And so if we look at this animal, now it won't have IgE antibodies, which means that it won't be able to have degranulation upon a peanut exposure, and that it would lose the signs and symptoms of anaphylaxis. But there's a twist here, which is that when this was done, that wasn't true. So they generated IgE deficient mice. They tried to sensitize these mice. And what happened was there was a compensation effect. When the mice didn't have IgE, something shifted. And now there was more IgG. And now that IgG is what's causing the anaphylactic response. So what this says to me as an immunologist, as somebody who wants to understand how these mechanisms work with the ultimate objective of finding therapies and cures down the line, is that we think about the immune system like this, like Rambo, where it's one cell, and that one cell is doing everything, it's fighting everything. We get rid of that cell, and then everything will stop. And in my mind, the reality is a little bit different. It's more like this. You see these extremely complicated diagrams in most immunology papers, which to all of the folks in medicine here, I apologize for that on behalf of my colleagues, because these things are insane. You can see how if we remove one little part of this, there's still a whole web of other things that are ongoing. And so it doesn't just have this on or off. There's often compensations and shifts and changes. I think of it more like a web of interacting cells that are overall coming together to make a response happen. And whenever I see these diagrams, the only thing I think of is a plate of spaghetti. They're just useless. There's things pointing everywhere, arrows pointing everywhere. And so my job as an immunologist kind of going into the future is thinking about, well, how can we untangle this plate of spaghetti to understand how it actually works? And there's a new toolkit for this. And this is where kind of my lab and what I'm hoping to do as I go into the future, this is where we're placing our efforts now. And this is in the domain of something called systems immunology. So the concept of studying the immune system or studying anything as a interconnected web or an interconnected system using technologies that match that concept. So rather than saying, here's one thing we're interested in and let's get rid of it, we're saying, well, let's look at how all the different cells and molecules come together to exert an effect. But this is a difficult thing to do, and it's difficult to wrap your head around. So I'm going to kind of talk to you about the cycle of systems immunology and how we think about doing this research. And so I think where a lot of this stuff starts is in some observation or some context in humans. So in this case, this is taken from a review by Bali Plender and Mark Davis from Stanford. And this is a human vaccination context, but you can just as easily say allergy or any other context for that. And you take some sample from this context. And then we use tools that allow us to survey lots of different parameters and in lots of different cells. And when you do that, you generate huge amounts of data. You can think of it as an Excel spreadsheet where the rows are different cells and the columns are different parameters. And you could have spreadsheets with hundreds of thousands of rows when you do something like this. And I'm not just sitting there and scrolling through and trying to find something in these data sets. So, one example is, these different data sets that are here. So transcriptomics, we can see for each individual cell, all of the RNA that those individual cells are making. And that sounds like a mind-boggling venture, and it is. It's because it is. But the way that we make sense of that in today's immunology is using some computational tools, using computers. And we can integrate these different parameters and even pull in clinical outcomes or clinical information and use that to basically crunch this data down into something that's useful. And when we do that, we output something useful. And this something useful can be used to generate new hypotheses. So we start at a place where we know that there's some interest, immunized versus not, allergic versus not. And then we can pull apart and start to understand what the data is telling us, and then we can generate hypotheses based on that. But what that looks like is going back into our mechanistic toolkit. So basically going from these kind of systems levels discoveries and moving into a knockout mouse or CRISPR knockouts or something of this nature. And I call this a systems to mechanism approach, which is really how I see my lab moving down the line. We come up with a hypothesis, we test that with the knockout, and then it is our duty to turn that into something useful. I think that's our moral obligation as basic scientists. And so, understanding things for their base is really also morally valuable, but some, if not the individual lab themselves, but somebody needs to take that information and turn it into some new biological insight. But the reason that this is a circle is because now you have a new intervention. And you can apply that intervention to people, and you can take samples, and you can start this cycle over and over again. And intersecting these things are things like clinical trials or other discovery-based science that can feed into new hypotheses. So this all sounds great, but I want to talk to you a little bit about how I managed to, or how we have applied this in action. So this systems to mechanistic approach. And something that we knew in the allergy space is that we have memory B cells. These are cells that make antibodies, or they have antibodies on their surface. And these memory B cells, when they are exposed to an antigen, they'll become a short-lived plasma cell. So they'll start making a bunch of IgE. And it's that replenishment that seems to keep people having these IgE titers, say, against people. But something that my peers and that even Ardner had shown previously is that these IgE memory B cells, they don't really exist. So the way that we have this memory for an allergy is through a different kind of B cell, which is an IgG memory B cell. And these IgG memory B cells will switch to become these IgE plasma cells. So there's kind of like a repertoire that changes and becomes these plasma cells. And it's this iterative cycle of making more IgE plasma cells that keeps these humoral titers. And I can't normally, if I was in person, I would say, does that make sense? But unfortunately, I don't think I can get some feedback if that makes sense here. But this was kind of the concept. But we didn't really know very much more than that. So we had a very simple question. I like to try and ask as simple a question as I can using these tools. And so we wanted to know what type of memory B cell holds this IgE memory. So we know it's IgG, but is there a specific type? And so to do this, you can think that we started at the circle. We found a context. We went, well, we have birch allergic people, we have some house dust mite allergic people, and we have some non-allergic people. And here's what we'll do. We'll take their PBMCs, we'll pull out their memory B cells, and then we'll use one of these omics technologies, these systems level technologies, which is single cell RNA sequencing. So for each memory B cell, we can find the 10,000 some odd genes that those memory B cells are transcribing. And then we know what different kinds there are in there using computational tools. And our dream, our hope, was that we would find a type of memory B cell that was in allergic people, but not in non-allergic people, something that we can say, this is the thing. So this is the output of some of this computational analysis. We have all these dimensions, different parameters, and we crunch it all down into something that we can plot in 2 dimensions. So what this is showing here, each dot that you see is an individual cell. And these individual cells, their position on this graph, which is called a UMAP plot, their position is dictated by all of the 10,000 things that they make. So the dots that come together here are cells that are similar and making similar things. And the ones that are further apart, well, there's some argument about this, but the ones that are further apart are more dissimilar. And so what we found was that their memory B cells are quite diverse. There's many different little groupings that you can make of memory B cells. But when we took a look at this in allergic and non-allergic people, we pretty much found that every single one of these clusters was represented in both non-allergic and allergic people. And this is just a percentage of the total cells that shows that there's no real difference between allergic and non-allergic. So our original hypothesis was not true, that we would find a specific cell that would be only in the allergic people. But the power of these systems tools is that that's not, we could have said, that's it, that's the end, we failed. And unfortunately, with the mechanistic toolkit, that's often what happens. You make a knockout mouse, and then it doesn't do anything. And you go, okay, well, that data will never see the light of day. But because in this systems toolkit here, we have generated so much data, there are things that we can find within this data. And what popped out to us right away was this grouping of cells that seemed to express markers related to allergic immunity. So, Fc epsilon receptor 2 here is a receptor for IgE. IL-4 receptor and IL-13 receptor. IL-4 and IL-13 are really the hallmark type 2 immune cytokines. And here, this says IgHE, which stands for IgE. And I did just get done telling you that there's no IgE memory B cells. And it turns out that these IgHE that you see here, it's not that they're IgE memory B cells, but they're cells that are transcribing the IgE and they're ready to become IgE B cells. So we have these IgG B cells that are actually ready to become IgE. And this hadn't been reported before. So we were really excited. But a problem, so what we did here, rather than trying to dive into this, we defined some different groupings that other people had described, CD95, C11C high. These ones tend to be involved in, like vaccine responses and in autoimmune conditions sometimes. And then we had here, which are like, unswitched naive cells. And then these 2 clusters here really were our MBC2 clusters, is what we called them, type 2 or allergic memory B cells. And so we use this as a means of trying to understand how these cells differ from all the other ones that are around. And I'll spare you a lot of details on this, but basically they differ because they make more stuff that is associated with allergy. But just because they're there, it doesn't necessarily mean that they're involved in allergic conditions. So the next thing that we needed to do is figure out if we find the B cells in people that are responding to an allergen, are those B cells this MBC2 phenotype that we have observed? And so to do this, we leveraged another thing that we developed in the lab that we published in January of 2024 in collaboration with Justin Taylor, who is at the University of Virginia, who's was previously at the Fred Hutch Institute, a very good friend of mine. We built these, what we call tetramer reagents, that basically are a peanut protein that have a fluorescent probe on them. And we can put that on our B cells, and the B cells that are responding to that specific antigen, they'll capture those probes and we'll be able to see them because they fluoresce. And so what we did was we grabbed some birch and some grass specific B cells and we put them through a different omics pipeline. And we were able to integrate that in with our omics, with our map that I just shown you. And so what we found when we did this was all of the cells that we found that were specific to birch or specific to grass in allergic people they clustered in with these MBC2 cells that we had observed. But when we looked at cells that were specific for the coronavirus, which is a viral infection, which, for the immunologist out there is really like the opposite of allergic immunity, we found that those cells didn't really fall within this green cluster here, although some of them maybe fell within the blue cluster here. So we found this really unique phenotype. This MBC2s were really present in these grass and birch allergic people, which really to us drove home, hey, this is associated. And we did some other work where we were able to make a connection between IgE plasma cells and these memory B cells, which really solidified that connection. And so we used this system immunology approach kind of coming around the side here. We had a condition, allergy versus not. We applied some systems immunology, and we got some bioinformatics analyses that found this new type of memory B cell. But we kind of have to come full circle on this, so we started generating some hypotheses. We needed to get mechanistic now. So we hypothesized that these memory B cells would require the type 2 cytokine IL-4 for being produced. But in order to do this, we needed some control that we couldn't really have in humans. We needed to get mice that didn't have IL-4. So the first thing we did was we jumped into the mouse and we found a set of cells that look very similar. So we did this in a bunch of different models, but basically what I'll show here is CD23 is that IgE receptor I was telling you about, and IL-4 receptor alpha here is the IL-4 receptor I was telling you about. And in the allergy context, we can find B cells. Each dot here is one B cell, and it's charting these things against each other. We can find B cells that are positive for both. But in the viral infection context, we didn't see the B cells that were positive for both. So we were able to identify a similar population. And then what we did from there was we jumped into this knockout mouse. So we got some IL-4 deficient mice. We tried to make them allergic, and we looked at their allergen-specific cells. And when we did that, the wild type, as I showed you before, looked like this, but the IL-4 deficient really didn't have any of the cells up in this gate, which we were able to use this mechanistic approach to find one of the key regulators of making these cells. So the next question that I have, as I'm kind of rounding up to 20 minutes here, is, why should you all here in a, grand rounds for medicine care about this? Which I think is because this is, in my mind, the direction that research is going. The tools for these omics approaches has substantially improved, even just in the relatively short time by comparison to my colleagues that I've been doing this type of research. And they're really good to a point where we can look at many cells with a lot of different parameters and in many different ways. And because it's moving so quickly in this direction, people have been applying it and finding things that we didn't know before. And they're doing it in a way that is kind of unbiased, doesn't require hypothesis. It's discovery research from which we distill hypotheses. But importantly, because I'm sure that I may have stricken some fear into the hearts of very basic scientists here, both the systems and mechanistic toolkit really matter. You need both of these things in order for success, because you can use this systems toolkit to find new things, but you don't know that those new things are really contributing to the disease process without diving into some model systems to get some mechanism. Is this actually important, what we found? So really, you need to have both in a kind of modern and functioning lab, in my opinion. What I'd really hope that people take away from this as a clinician and, even as researchers is, to keep an eye out for human contexts. This could be, things that you're seeing in the clinic that people maybe haven't been reported, and maybe we can apply some of these tools to make some discovery here. More related to my work, there seems to be a new type of memory B cell out there, a type 2 polarized memory B cell. And, we are ambitious in thinking that this has a role in a few different diseases. And I think we've, from a clinical standpoint, coming back around, we know that IL-4 is important for making these cells. So I think that there is some stronger evidence for a curative potential for dupilumab in allergic diseases, which I know is already being pursued and approved in many allergic contexts. But in food allergy, what we study, still not approved. And I think that, Novartis has this in trials. And I think Regeneron has this in trials. And I think it's really worthwhile. So, I was trained here at McMaster by Dr. Manel Jordana, who is an incredible mentor. and Susan Wasserman, who's an incredible mentor as well, who I saw in the audience here. And really, I couldn't be the scientist I am today without these two people. And they've given me really everything that I have. And so a lot of this should be attributed to them as well. And, here's our funders. Here's some conclusions for you. And we dressed up as pipettes for Halloween last year, which was really fun. And this is Dr. Jordana as a pipette, just in case anybody needed that in their lives.
Thanks, Josh. That's really excellent. I think one of the things that I wanted to feature, and you've done it really well, is the fact that we do have people doing discovery research in the department. Oftentimes, people think of the Department of Medicine as a big clinical trials research engine, systematic reviews, meta-analysis. But in fact, as I said at the start, we have almost 50 fundamental scientists working in discovery research or in taking observations, as you just said, from the clinic back to the lab, which is an area where there's immense interest. The other thing that I've been really focusing on for the last couple of years is innovation and commercialization, and how can we take some of the great ideas that you and your colleagues have and move them into applications in humans, which is an area of a lot of interest and focus. So thanks for that really great summary of your research in 20 minutes, which is remarkable. And I open up for any questions. You can either ask in the webinar chat or in the question and answer. There's one comment from Imran Sachi just saying, wonderful summary of all your hard work. So congratulations.
Thank you.
And I'll just kick off a question while we're waiting. So, one of the things I've learned over the last little while is that allergy is way more complicated than I think of it. And it sounds like you would agree with that. Are there any fundamental underlying principles? Why do some people get allergies and others don't? Why does the day that the ragweed starts flowering, does my life descend into chaos?
This is something that I'd really love to be able to give you a nice and clear answer on. I imagine there's a lot of people here who do, epidemiology work. One of the real tricky parts with this type of work is that people show up to the clinic already allergic to stuff. And so we don't have a categorized list of everything that person has done before they came to the clinic, right? So it's hard to find these associations. But I think that the boom in the genomic space, where we're able to get cheaper and cheaper genomes, I think we are going to reveal that a larger number of people than we had anticipated have these issues because of some inborn error. But I think that can't encompass everything. So there is probably a genetic component to it, but I think that there's also a context component to it. I think people are exposed in a way that is not that is unflattering to the allergen, and then the immune response kicks in and goes. So I don't have more. Unfortunately, that's a very crappy answer to a good question.
Thanks. And we'll come back later to tell people how the Black Death influenced the prevalence of allergies in
yes
in the population.
I need to talk to Henrik. Yeah.
There's two questions here. One is, could memory B cells sometimes be developed to have anti-tumor effects?
110%. Completely right. Right now, we're kind of in a boom of understanding that B cells are a huge player in immune surveillance in cancer. So basically, in most cancers, if you have, germinal centers or B cell infiltrates that are inside the tumor, you're going to have a better outcome than people who don't. There are exceptions to that rule, but by and large, it's true. And we're learning that there are targets that antibodies see. We don't know exactly how they work, but we do know that B cells have a huge role. And something that people are doing is actually, engineering B cells to have an antibody target for a neoepitope that's on a tumor. And then, reinfusing those in people like CAR T cells, and those antibodies are going and helping in the protection against the cancer. So I think this is a huge field, and I think it's something that everyone needs to think about some more.
Thanks for that. That's great. And Imran asked a question in the question and answer, and I'm going to muck up the name of the drugs. Robert Wood just published the impact of omalizumab on multiple allergies in the New England Journal last week. Your thoughts on this data, and would Dupi, which I presume we mentioned earlier, would be more effective?
So Dupi is a monotherapy. It didn't reach primary endpoint on clinicaltrials.gov. So I think that as a monotherapy, dupilumab's not going to be that helpful, which is not a huge surprise because memory B cells tend to be pretty quiescent. So yeah, the IL-4 blockade can be there, but if the cells aren't activated, nothing will happen. A really interesting thing is that Regeneron has not released, even though they've completed the trial, they have not released the outcome of their therapy with the allergen in the form of an oral immunotherapy, which I'm sure Susan Wasserman, who I think is here, could talk a lot more than I could about. But I think that there's a lot of potential in that, and there's a few papers to come out recently. The omalizumab work is very interesting, but we know it's not curative. We know that people who stop taking omalizumab will just make more IgE. So I think it's great, especially as, and Susan tells me there's a lot of people with extremely severe multi-allergies where going on immunotherapies is not a huge option for these folks. And I think omalizumab in that context will be a very helpful drug.
Excellent. So Josh, thanks very much. Josh has to run off to another meeting. So if you can unshare your screen, that would be great.
Yeah, absolutely. Thanks so much for inviting me. It was fun.
Yeah, no problem. If Dr. Rullo, if you're there, if you want to share your screen, and we'll swim over to Dr. Rullo, and I'll give him a quick intro while he's figuring that out. So Anthony Rullo joined us a couple of years ago from the Boston area, although he's from Hamilton originally. And Anthony does some really exciting work in covalent chemistry, which he's going to talk about. And Anthony, I will tell you that we can see your slides properly. So Josh, if you want to turn off your camera and your mic, just in case you're going to disappear, and I'll turn off my camera and mic. And Dr. Rullo, go ahead.
Thank you very much. Can you guys hear me okay?
Yeah, you're fine.
Great. Pleasure to be here. Thanks a lot, Josh. That was fantastic. Now we're going to talk about something a little bit different in the therapeutic covalent drug space. The title of the talk today has to do with work our group's been doing here at McMaster over the past 6 years, where we try to develop new therapeutic modalities for disease. A lot of our initial applications have been in oncology, where we develop a drug, which is depicted here as B, this green bifunctional molecule. And in contrast to more traditional drugs that are developed to bind a target and inhibit a function, where B binds A, let's say, in our case, we're trying to bridge an additional species to recognize the target. So B, this bifunctional molecule, actually bridges another protein in your body, C, with a target protein A, okay, in the most general terms possible. And this interaction is dictated by two binding constants, okay, KAB and KBC. And so this, in a nutshell, pictorially describes the concept of proximity induction, bringing two things in close proximity to enforce a pharmacological and therapeutic effects, okay? So just a general introduction to this proximity induction concept. This all started actually with the Stuart Schreiber at the Broad Institute coining this term proximity-inducing molecules, these new types of drugs that don't just bind a target, they actually recruit additional machinery in your body to recognize that target, okay? And this is based on the concept of increasing what's called effective molarity of interacting species. So you're bringing things close together, which basically is like simulating a much higher concentration of those two species than there really is. An example is kind of, the really high protein concentrations that are in the endoplasmic reticulum, okay? And you can get all kinds of functions at these concentrations that you would not get under more dilute conditions. Getting them close together is only part of the battle. You also want to get them close together in the right orientation so that they interact properly. So for example, your antibody is recruited to your tumor, okay, in the proper orientation to elicit opsonization. That's an example. So this was founded really in chemical concepts. I'll just give you a very quick overview here, where you have this reaction on the left that happens between two reagents. I'm not sure if you can see my cursor here. This carboxylate attacks this acid chloride in a very slow reaction to form this anhydride. This is kind of bringing you back to your beloved second-year organic chemistry classes. If you do this exact same reaction, but now you tether the reactive groups in one molecule, so this is now an intramolecular reaction, and it happens much faster because you've paid the entropic cost of bringing things together. You've increased the effective molarity of your reactive groups, okay, to get a chemical reaction. If you further pre-organize these in this more strained Norborian type core, you get an even faster reaction. So this is illustrating the concept of proximity induction from a chemical perspective, okay? In a biological perspective, we see this all the time. We see this with transcription factors. Here's an example here with these Z scaffold proteins. So in the absence of this STE5 scaffold protein, These STE kinases interact with each other with very low kinetics, okay? But if they can all bind the scaffold protein, they pre-organize. The entropic cost of interacting is paid for by the scaffolding protein. And now the reaction rate is much, much faster, okay? So this is the concept behind the development of these new therapeutic modalities that we're calling proximity-inducing molecules. Some of you may have heard of PROTACs, okay, which mediate intracellular degradation. In immuno-oncology, you may have heard of bispecific T-cell engagers or BiTEs that bridge CD3 epsilon with a tumor antigen. Okay, these all fall under the category of proximity-inducing molecules. And the goal of these drugs or modalities, again, is to increase the effective molarity of biological species to drive a functional outcome. Okay, this is beyond occupancy-driven pharmacology like antagonism, where you make a drug that inhibits a receptor's ability to bind its natural ligand. What we're doing now is we're actually targeting things that are typically undruggable for a variety of reasons, okay? And we can access biological clearance mechanisms like immune elimination or proteolytic degradation. An important concept in these types of drug modalities is that instead of a binary complex where the drug binds the target, these require a minimal ternary complex. So you've got three components binding together. Okay, now this has really important consequences. And this is something that our lab has worked to really optimize to get these proximity-inducing drugs to work better. Okay, so to understand this a little bit more, I'm just going to show you here this concept. This is the original depiction of a proximity-inducing molecule B that bridges, let's say, a tumor antigen A to an immune component, let's say an Fc receptor or an antibody, okay, C. And the three binding form a ternary complex. Now, the tricky thing about these types of drugs is that their binding equilibria are much more complex. Without getting too much into the physical chemistry here, I just want to illustrate that there's at least two binding constants for you to form your productive ternary complex, which is shown here as ABC, okay? Your drug B can bind the tumor antigen A first and go along the top right pathway, or it can bind your immune component C first and go along the bottom pathway, all right? Now, what makes these tricky to develop is that it's hard to maximize your ternary complex. It's hard to have two really high binding affinities where you're favoring the three coming together to form ABC, okay? In the context of BiTEs, this means it's really hard for you to have the BiTE enforce bridging of the T cell with the tumor, okay, because of these physical chemistry restraints. If you don't have something called positive cooperativity in your system, it's really difficult to maximize this ABC, okay? And positive cooperativity just means that when your drug binds A, it now binds the protein C with higher affinity, okay? And that does not happen in the majority of biological cases. Okay, you do not get positive cooperativity. In fact, you get something that's non-cooperative or what's called negatively cooperative. This actually means that there's natural thermodynamic factors that are limiting your drug's ability to bridge the two components of interest. Okay, one example are these molecules that we actually were developing called ARMs or antibody recruiting molecules. Okay, and here's an example of what I mean. The antibody recruiting molecule, shown as a blue sphere, is administered and it's rapidly cleared from the body, which is one problem. Now, for it to work, it has to bind serum antibodies in your blood, okay, shown here, this first equilibria, and has to also bind to tumor antigens on the cancer surface through the second binding constant to form this ternary complex, okay? The ternary complex here that's required for function, this ABC notation, is our bifunctional arm, or bispecific, you can think of it that way, bridging serum antibody with tumor antigen, which leads to FC-dependent effector function, okay? And so a big problem here is that we need really high affinities to get this, to drive this process towards bridging of the two species to get the therapeutic effect, highly sensitive to binding affinity. And also we have this something called negative cooperativity that makes this even less favorable because ultimately in immuno-oncology, you're trying to bridge immune cells with tumor cells. Okay, so there's a lot of forces that are resisting that and reducing our immuno-oncology therapeutic efficacy. Okay, this here is a theoretical dose response curve for a proximity inducing molecule like a BiTE, okay, or like an antibody recruiting molecule shown here. In both cases, you want to form ternary complex ABC, okay? So you increase the dose of your drug B, and you get an increase in your functional ternary complex, all right? The problem with ternary complexes is that you get a bell-shaped curve in the pharmacology. As you increase the concentration of your drug, you start to actually auto-inhibit the ternary complex that you require for function, okay? Additionally, problematically with these type of drugs, if you don't have high enough binding affinities, you actually can never saturate your target occupancy. This is not the case for a traditional drug because even if the potency is low, you can keep increasing the dose and eventually you'll saturate binding. With this type of proximity-inducing drug, you'll never saturate binding. The peak of this bell curve will never be 100%. Not to mention, if your concentrations are too high, you'll actually be inhibiting it. Okay. In the context of bridging immune cells, let's say with tumor cells, there's another problem, this negative cooperativity I mentioned, where the shape of this bell-shaped curve actually contracts and gets lower. So now negative cooperativity, which is preventing you from forming the ternary complex, has made it even harder for your drug to be therapeutically active. And so these types of problems limit the efficacy of a lot of current proximity-inducing molecules, like PROTACs, like BiTEs, monoclonal antibodies that bridge immune receptors with tumor antigens also follow these same pharmacologies, okay? And so our group worked to develop something called a covalent proximity induction strategy that is designed to enforce ternary complex formation. So here's an example of something that we called a covalent immune recruiter, or a CIR. If you remember in our previous slide, we had the antibody recruiting molecules that were these blue spheres, these blue circles, that bind an antibody and simultaneously bind a tumor antigen to form the ternary complex. Well, here we have a similar molecule that has this red arrow, and what that red arrow is a covalent group, shown here is an activated acylamidazole ester. What that allows you to do is enforce binding to your target through covalent engagement. Now the bispecific blue molecule doesn't just bind to your protein, it covalently locks onto it. In this example, it covalently locks onto a serum antibody of your choice and localizes that to an additional protein receptor on a tumor antigen, in this application to enforce tumor opsonization and ADCP, ADCC killing of the tumor. But you can actually enforce proximity between any two proteins in the body that you might be interested in autoimmunity, in allergy, okay? Whenever you want to bring one protein to another protein, this is a chemical covalent tool that you can use to accomplish this. And the way the chemistry works, it's something called affinity labeling chemistry, where again, this concept of proximity is used, where you bring things close together, you increase the reaction rate so that you only get covalent reaction with the target protein that you're binding. That's a big part of what our lab develops. And so we discovered something really interesting with this in the context of immune activation through Fc receptors, okay? Where it looks like covalent engagement gives you an increase in immune function for reasons that actually are beyond forming more ternary complex. And this we were not expecting. On the left here is a flow cytometry bar chart. In the red, so what we're doing is we're measuring the amount of serum antibody that our bifunctional molecules can recruit to a tumor cell, okay? So the x-axis here, if we look at the red and blue bars, the ones on the left are controls, don't worry about that. So for the non-covalent bifunctional, which we call NCIR, non-covalent recruiter, you can see here you increase the concentration of drug and antibody. And as you go from left to right, eventually you get a really high amount of ternary complex on the cells measured by fluorescence. So now all the PSMA tumor antigens are bridged with serum antibodies. It's actually a high affinity anti-DMP serum antibody that we targeted in this case. If you look in blue, this is when our bifunctional CIR can covalently engage the antibody. You can see here that at lower concentrations of antibody and drug, we get a higher amount of ternary complex on the cancer cell surface, more antibody recruitment, higher signal at lower doses. But importantly, at high concentrations of the drug and the antibody, We saturate all tumor antigens with bridging antibodies. So in this regime, we'd expect them to have similar efficacy. When we do the exact same experiment, but now we look at Fc receptor activation, so this is CD16 on natural killer cells, okay, in a luminescence ADCC assay. We do the same titration. In black is the ability of the covalent molecule, the CIR, to activate CD16. So when antibodies are recruited to the tumor, CD16 is cross-linked and activated, okay? Dose-dependent increase in function, which is great and akin to a therapeutic antibody. These assays are used to look at monoclonal antibody, ADCC. Interestingly, though, even at these high concentrations where both are covalent and non-covalent, gave us equal amounts of ternary complex on the cell surface, equal amounts of antibody recruitment, the non-covalent bifunctional has actually no efficacy at all. And this we were not expecting. You can see it's in the baseline. So the covalent bridging of the antibody to the tumor cell increased Fc receptor activation for reasons beyond receptor occupancy, because we know the non-covalent version was still making the same number of bridges because it's a very high affinity binder. So at this regime, we have comparable amounts of ternary complex, but we have such different levels of receptor activation. So that is something that was really intriguing to us and something that we think has enormous implications in immuno-oncology and even receptor pharmacology. This actually led to 2 current models that we're investigating in collaboration with the Bramson lab, actually, and I'll show you a couple slides on that to end things up shortly. One is this idea of a residence time. So it's not just enough that you're bridging your 2 proteins together. The idea is that they have to be bridged for a certain amount of time to commit a certain signal or response. So here the blue molecule is our bispecific. It bridges tumor antigens with antibodies, okay? The antibody is bound to Fc receptors, which have to cluster for you to get ITAM signaling and function. So it's not just enough to saturate these bridging interactions. They need to be held for long enough for you to get clustering and signaling leading to an immune response. Any microscopic dissociation might reset this process. So for that reason, K-off or residence time is very crucial, and covalency infinitely increases that residence time. The second model we're working on and thinking about is this mechanical strain model. The idea here is that there are natural mechanical forces that are actually pushing the cells away from each other, kind of this negative cooperativity concept I mentioned earlier. So this bridging complex that you're trying to enforce with your therapeutic is actually being ripped apart by these mechanical forces, for example, between an immune cell and a tumor cell. So the problem here is even when pharma engineers a high affinity bridging molecule, like a biologic or a BiTE, the problem is that this mechanical strain will actually disrupt that, even if the affinity is high. But covalency prevents them from coming apart. And so we think that this might be another mechanism that's happening. But the take-home message here really is that in these tumor immunotherapeutic applications, the function is not simply governed by how many receptors you're binding. It has to do with this kind of residence time or this covalent engagement. So we thought that was really a cool discovery with these tools. I'm just going to finish off by showing you another system where we basically saw the same thing. So instead of looking at Fc receptor activation with these covalent bifunctionals, we looked at T cell activation in collaboration with the Bramson lab, where together we developed universal receptor T cells. So the T cell is now engineered with a universal receptor. And this receptor, instead of recognizing a tumor antigen the way that CAR T cells do, it recognizes our bifunctional molecule, shown here by a cartoon with a red circle that binds to the receptor on the T cell and a green triangle that binds to the tumor antigen of choice. This could be HER2, this could be folate receptor, etc. And so these are our same bifunctional small molecules, and now they actually bridge the T cell universal receptor with the tumor antigen. And again, here we compared covalent bifunctionals that lock onto the T cell receptor the way they did before with serum anti-DMP antibodies, and compared function to when the same bifunctional was used, same binding affinities, but no ability to covalently engage, no reactive group, no electrophile. We actually did this with 3 different synthetic antigen receptors, DNP-TAC that the Bramson labs developed, HER-CAR, and the traditional DNP-CAR. These are associated with very different tonic signaling propensities, with CAR having the highest. which is attributed to its off-tumor toxicity. On the next slide here, just finishing up, what we saw was that, again, covalency makes a huge difference in immune activation. If we look on the left, the DMP TAC, we look at cytotoxicity against LNCAP tumor cells as a function of dose of our bifunctional molecule, whether it's covalent or not. CIR in blue is covalent, and CIR in red is the same but non-covalent. You can see for the TAC, which has very little to no tonic signaling, so it's almost like it has a higher activation threshold. The non-covalent molecule, even though it's high affinity and it can bridge the two, it's not activating it, but the covalency does. Okay, and there's a significant difference between the two. In the intermediate case, where this has intermediate tonic signaling, covalency is really key and gives you a huge difference between the non-covalent. And this is at doses where the non-covalent is bridging the two cells and receptors just fine. Interestingly, in the case of the CAR, where you don't have this high activation threshold and you have very high tonic signaling, covalency helps. It gives you a potency shift, but you don't see this difference in activation. Okay, so it's really interesting. It really tells us something about the pharmacology and the activation thresholds. But again, this concept that covalent proximity induction as a drug, when you're bridging two proteins, can really increase the efficacy of the drug. For a variety of reasons that we're exploring in the models that I had mentioned. And this data was confocal microscopy data done with the same immune receptors, under the same conditions. And this was done to see if the covalent molecules were working better because they were bridging more cells. But as we hypothesized, they were not. If you look here in the case of all three receptors, the CIR in blue and the non-covalent analog in red, they both form the same comparable number of T cell tumor cell conjugates looked at by microscopy. We wanted to see if maybe the covalent groups, the covalent bifunctionals, were increasing immune synapse formation, which we measured by looking at CD45 phosphatase exclusion. Okay, as you can see here, we could image the tumor cell in red with PSMA and cherry, and we could look at the T cell CD45 phosphatase in blue, and we could look at the synapse. And again, in all three receptor cases, there is no difference in CD45 exclusion and in phosphatase exclusion or synapse formation. So the contacts are the same. It was when we looked at markers of T cell activation that we really saw the difference. When we started looking at a little bit downstream of synapse formation, we started looking at phosphorylated ERK. Now we're seeing in the receptors in the T cell receptors where we saw the biggest functional increase from covalent engagement, now we're seeing a very big difference in signaling. So covalency in blue, a significant increase in phosphorylated ERK when the bifunctional is covalent versus the non-covalent. Same with the intermediary tonic signaling receptor, the CAR, okay, and very little difference with the CAR. So, and this actually translated even further downstream in the T cell activation pathway. Here is a little schematic. It binds the T cell receptor, and then you get phosphorylated ERK, this NUR77 expression and CD69 expression. You can see that they are very elevated when the bifunctional is covalent on the left here in blue versus non-covalent in red, even though the number of contacts are the same. So in immuno-oncology applications, we see this real increase in function from proximity-inducing molecules that can covalently bridge the two species together. Okay, so that's really the conclusions of what we're doing here. We're looking at direct covalent engagement of immune cells like endogenous macrophages, tumor-associated macrophages. We're developing these molecules in hematology to try to knock out autoantibodies and working on this universal T cell platform. So I just wanted to thank you all very much for your attention. Thanks a lot, Dr. Crother, for bringing me out here today. I want to thank our colleagues and collaborators, especially the Bramson 1 and Singh labs at McMaster. This is our current group here on the left, and my daughter's on the right here. Thank you very much.
Excellent. Thanks, Anthony. That was really great. I'll just ask people if they have any questions to put them in the chat or in the question and answer. And Mark's got one, but I'm going to ask you one first. So you and I have talked about this a lot. There's a lot of excitement about the work that you do. It's really a burgeoning field. Just for the clinical people, can you give them kind of a concrete example where you think this technology is likely to go, a clinical application within which bringing these molecules together is going to hit the clinic first? Not necessarily in your own area, but in a general sense.
In a general area, I think that these types of concepts are starting to work in protein degradation. That's where it's getting into the clinic right away. So the kind of in the PROTAC space is where you're starting to see these bifunctional proximity-inducing molecules can really lead to exciting therapeutic outcomes. So you're seeing PROTACs in oncology. I think that you're going to start very soon. You're going to see in autoimmunity, you're going to see that you can actually deplete and target extracellular proteins for liver degradation or for depletion. So you can get rid of a lot of autoantibodies and other factors, maybe also probably in some certain blood diseases. I think you're going to see that you can bridge target these proteins for elimination or degradation outside of immuno-oncology.
Yeah, I would agree with that completely. I'm remotely involved in a project with a major pharmaceutical company in immune thrombocytopenic purpura, or ITP, which is an immune-mediated disease where they're looking at technology like this to deplete immunoglobulins, which presumably are in some way an effector of that disease. So the early phase work is really quite exciting. So it's quite neat.
Yeah.
This is an entirely new area of science, but it also could allow us to recycle some of our older drugs, I think, that have effectively lost their appeal because newer ones that are more safe and more effective come along, but this could allow us to better use some of the older drugs we've got.
Yeah, exactly. Yeah, and there's a lot. Pharma has a lot of drugs that were never potent enough to inhibit or antagonize a function, but those ligands, if you incorporate them into a bispecific molecule and you add covalent binding properties, now can become very efficacious proximity-inducing molecules.
Yeah, totally agree. We've got two questions here. So Mark Larche, a bit of a long question. Excellent science, Anthony. Clearly lots of potential to overcome the problems of the two-step progression to a ternary complex. Would it be possible to make a recruiter that has cryptic affinity for one of the targets, for example, by virtue of a repelling charge in the original molecule or a hidden allosteric property that is not lost or unmasked until the first target has been bound?
Professor Larche is so brilliant. So the idea is that, did I follow this right? That you can control the order of bridging? That you would only bind the second one once you bound the first?
I do not know the answer to that other than to say it looks like he is trying to eliminate a two-step molecule. He's saying yes. He put yes.
Yeah, that's actually something that we're working on. I think that your charge idea is a very clever one. And I think there's a lot of potential in that direction and controlling the order of bridging, because once you can control the order of bridging, you can not only enforce the ternary complex, but you can also redeploy those same types of molecules to do things like, release a chemo drug only when it binds the tumor. You now have like all kinds of demasking strategies and things that you can devise from that.
Perfect. In fact, that's a nice allusion to the next question from Imran again. Fascinating talk. One of the problems with novel immune therapy for cancers is serious side effects such as pneumonitis or hepatitis, so-called off-target effects. Can these proximity molecules be used so that biologic effects or side effects are limited to the target organ? I think you just answered that, but more detail on that?
So I guess if we're talking about the type of off-target toxicity of, let's say, like a BiTE or a bispecific engager, the approach that I talked about today is really meant to increase efficacy. It could reduce toxicity by working at lower concentrations, but this is not what I talked about today is not designed to reduce the toxicity per se. That would be a separate question to address. So,
but could you target? Could you target it only to the impacted cell and thus avoid off-target effects, say in the liver, where right now the off-target effects are in part just because the liver is vacuuming all this stuff up?
Oh yeah, you can definitely put masking groups on so that it doesn't go to the liver, right? If your targeting domain works very well, then it should partition to the target before it gets to the liver, right? There's ways to kind of change the biodistribution for sure. Yeah, it's a smart question.
Excellent. Okay, well, we've got just a couple minutes left. I'll just draw this to a close, I think, since we are out of questions. Thanks, everybody, for listening. Thanks to Josh and to Anthony for really exciting insights into some of the basic science research going on in the department. Again, medicine is a big basic science department. People forget that, and it's really great to have examples from some of our younger scientists who are doing some really exciting stuff, moving science forward and providing the deep insight that we as clinicians use when we're treating our patients. So thanks, Anthony. Thanks to Joshua who had to run. And also thanks to the listeners. If you have questions or concerns, you can send me an email. And this will be posted on our YouTube channel if anybody wishes to watch it later. Thanks very much, everybody. Talk to you later.